So I should say that I may not look at the chat that much, I may forget. I'll try my best. So if you have a question, just unmute and ask me, okay? So this is glycogen metabolism, but I would like to start off with sort of a big picture slide, just because some people get confused with biochemistry and they find it difficult to understand regulation, which enzymes are activated by insulin, which enzymes are activated by glucagon, whether those enzymes are activated by phosphorylation or dephosphorylation. And so this is just a general approach which I use to make it simpler for myself. The concept is that the body likes homeostasis, right? It needs to maintain certain parameters within a set limit or at a set point. The same applies for blood glucose. Your body likes to maintain blood glucose within a certain limit, about 80 to 110 milligrams per deciliter. That's the set limit of glucose. So if glucose goes up, the body will work to bring it down. If glucose goes down, the body will work to bring it up. When you eat a meal, glucose goes up, right? So your body releases a hormone called insulin, and insulin basically decreases blood glucose along with the principle of homeostasis. Glucagon is released during fasting conditions. During fasting conditions, blood glucose falls down, and your body wants to maintain that blood glucose because the brain and the red blood cells only rely on blood glucose. The brain can rely on other stuff when it's prolonged fasting, but usually the brain and red blood cells utilize glucose because you remember the red blood cells don't have mitochondria, so they can't use oxidative phosphorylation or fatty acid oxidation for energy. They only use glycolysis for energy. So that's why the body wants to maintain blood glucose. So the body releases glucagon during fasting conditions, and glucagon maintains blood glucose. That's the first important thing you need to realize, that the body, when it's well-fed, releases insulin, and the pathways which are associated with insulin are active. When the body is in fasting, the body releases glucagon, and then the pathways which are activated by glucagon are active during fasting. That's the first point. The second point is insulin. Let me right. So that's the first important point. The second point is insulin acts by dephosphorylation. Insulin is a water-soluble hormone, so it binds to its cell surface receptor that activates a signaling pathway, and that causes dephosphorylation of enzymes. Now, depending on what that enzyme is, it could be active in dephosphorylation or inactive. If it's an enzyme in a pathway which uses glucose, which is the goal of insulin, is to use glucose, that enzyme will be active. If that's an enzyme participating in a pathway which makes glucose, like gluconeogenesis, insulin wants to inhibit that, so that enzyme will be inactivated by dephosphorylation. Okay, so again, so these are the pathways which are activated in a well-fed state. Like, so in a well-fed state, again, your body releases insulin, and the insulin uses up the glucose. How does it do this? It activates glycolysis. Do any of you know what enzymes insulin activates in glycolysis? What are the rate-limiting enzymes of glycolysis, or what are the enzymes which are activated by insulin in glycolysis? Okay, so insulin activates glucokinase in the first step of glycolysis in the liver. Insulin activates phosphofructokinase 2 in which is the rate-limiting step of glycolysis. And because I told you here that insulin causes dephosphorylation, these enzymes will be activated by dephosphorylation. Glycogen synthesis is activated in the well-fed state, so your body wants to use up glucose. So what does it do? Insulin tells the liver to utilize glucose by activating glycolysis. But eventually the liver's ATP stores are going to deplete. The liver is going to say, I have enough ATP. So insulin will activate glycogen synthesis again by dephosphorylation. But your body can only store a limited amount of glycogen. Your body can only store a limited amount of glycogen, and we'll discuss why later. But when the glycogen stores are full and the liver says, I can't store any more glycogen, insulin activates fatty acid synthesis. Okay, so these are the pathways which are activated by insulin. All of them are activated by dephosphorylation. Again, in the fasting state, your body likes to break down glycogen because glucagon is active. Glycogen breakdown occurs, then gluconeogenesis occurs, then fatty acid oxidation. Basically, glucagon goes to the adipocytes, tells it to release fatty acids for energy. Okay? So these are pathways that you should keep in mind if the body is fasting and if the body is active, if the body is well fed, sorry. Okay, so this is a summary. Glucose can be obtained from the diet. from glycogen breakdown or the production of new glucose from gluconeogenesis. When you eat a high carbohydrate meal, the pancreatic beta cells release insulin, and these are the actions of insulin. Insulin activates glycolysis in the liver by activating glucokinase and phosphofructokinase 2. Insulin activates glucose uptake into skeletal muscles and adipocytes by activating GLUT4. Insulin activates glycogen synthesis by activating glycogen synthase. This you will take in MOL 2, so I'm going to skip this. Okay, so let's move on to glycogen now, which is the topic. So I hope regulation makes sense to you. So glycogen is basically the storage form of glucose. The first question is, why should you store glucose? Firstly, because your body doesn't know when it's going to eat again. So it needs to store some energy when you're fasting. Okay. Second thing is, why don't we just store glucose rather than making it into glycogen? Because glucose, as you can see here, has many oxygen and hydrogen bonds, which means glucose is polar, which means glucose is osmotic. So if the body stores, if cells store a lot of glucose inside the cell, it will hold on to water, water will come into the cell, and the cell will swell up. So the body can't store a lot of glucose because it's highly, highly osmotic. So the body, what your cells do is convert it to glycogen, which is less osmotic, okay? Then the next question is, why polymerize the glucose? So this is the structure of glycogen. There's a protein called glycogen in the middle, and then you basically make glucose, you basically add glucose in a chain, all right? So the question is, as you can see, glucose is linked here by alpha-1,4 glycosidic bond. So one chain, if you add glucose in a chain, those glucose molecules will be linked by an alpha-1,4 glycosidic bond. So each glucose molecule here in this chain is linked by a 1,4 glycosidic bond. But then you have branches here, 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 and here. For example, these dots, these are branch points. These branch points are linked by an alpha-1,6 glycosidic bond. And this is important for you to know. So along each branch, you have alpha-1,4 glycosidic bond. At branch points, you have alpha-1,6 glycosidic bonds. All right. So the next question is, why do you need branched glycogen rather than just making straight glycogen in one chain? Well, with the branch structure, it's easier to store, firstly. And secondly, it's easier to break down. So an enzyme can act on this branch and break it down. At the same time, you can break down this branch, you can break down this branch. If glycogen was just in one chain, you would have to break down one glucose at a time. This way, you can, by making it branched, you can increase its rate of breakdown. All right. So that's the structure of glycogen. Branch structure, in a chain, glucose linked by alpha-1,4 glycosidic bonds. At branch points, alpha-1,6 glycosidic bonds. The next question is, where is glycogen stored? Glycogen is stored in most tissues, just a small amount, because when you're fasting, you need to break down glycogen for energy. So most tissues store glycogen themselves. But the major storage points of glycogen are the skeletal muscle and the liver. Okay. Now, this is a question for you guys to think about. Why is that glycogen is not the major storage form of energy in the body? Why do you think that is? We always learned that glycogen is the storage form of glucose, but most of your body's energy isn't stored as glycogen. Does anyone know why? Okay, so glycogen again is osmotic. So if you store a lot of it, it will hold on to water, and that will drag water inside the cell, and that will cause the cell to swell up. So there are diseases called glycogen storage diseases, like Pompe disease, where glycogen breakdown is ineffective, is abnormal. So glycogen accumulates in the heart, for example, and that causes enlargement of the heart. So your body likes to store a very specific amount of glycogen, right? So keep in mind, why do you form glycogen? Where do you store glycogen? In the skeletal muscle and liver. And keep in mind that glycogen is not the major storage form of energy in the body. This is just the pathway of glycogen synthesis. This is important. This is probably the most important slide. When you eat a high carbohydrate meal, again, your body releases insulin. Insulin causes uptake of glucose into cells. That glucose is trapped inside the cell by conversion to glucose 6-phosphate. Then an enzyme acts on it called isomerase. You don't need to know that enzyme. That converts glucose 6-phosphate to glucose 1-phosphate. Because glucose 6-phosphate has two options. It can either go into glycogen synthesis or it can go into glycolysis. So when your ATP stores are full and you don't need more glycolysis, your body converts glucose 6-phosphate to glucose 1-phosphate. to get put into glycogen synthesis. Now there's an additional step you need to know. For glucose 1-phosphate to be added to glycogen, it needs to be converted into UDP glucose. All right. This is the activated form of glucose. The enzyme which synthesizes glycogen requires glucose to be in this form for it to be added to glycogen. Right. So again, then what's the enzyme which mediates glycogen synthesis? For glycogen synthase. So glycogen synthase will add glucose molecules along the chain. So it will form 1,4 alpha 1,4 glycosidic bonds. For branch points, glycogen synthase can't form alpha 1,6 glycosidic bond. For that, you need branching enzyme, right? And if I ask you what hormone activates glycogen synthesis, you should say insulin, because glycogen synthesis would act when you have high blood glucose. You don't want to store glucose if you have low blood glucose. You only want to store glucose if you have a high blood glucose or if you're well fed. That means insulin is active. And again, I told you that insulin activates enzymes by dephosphorylation. And so if insulin is activating glycogen synthesis, obviously it will activate it by dephosphorylation, right? So for that's just glycogen synthesis. We're done with glycogen synthesis. There's 2 enzymes you need to know. Glycogen synthase for alpha 1,4 glycosidic bonds, branching enzyme for alpha 1,6 glycosidic bonds, glycogen synthase activated by insulin by dephosphorylation. For glucose 1-phosphate to be added onto glycogen, it needs to be in the form of UDP. Okay, that's glycogen synthesis. Now, when you're fasting or when you're exercising, your body needs to maintain glucose, and when you're exercising, your skeletal muscles need to make ATP, so they need glucose. So when you're fasting, your pancreas release glucagon, which acts in the liver. Epinephrine is released during exercise, and because exercise is a sympathetic response, a stress response, so epinephrine will act on the liver and muscle to break down glycogen to make sure that glucose is available for skeletal muscles for them to burn it and make energy in the form of ATP to keep those cross-bridge cycling. All right. AMP is another activator you should know. AMP is derived from ATP. And so if you have a lot of AMP, that means you've broken down ATP, right? So AMP is a signal of low energy. So when AMP accumulates inside the cell, that means the cell doesn't have enough ATP. If it doesn't have enough ATP, it wants to make more ATP. It can do that by glycolysis and oxidative phosphorylation, etc. For that to happen, it needs glucose. And so AMP will activate glycogen phosphorylase to break down glycogen to glucose for it to be then used in glycolysis and to make ATP. All right. So for glycogen synthesis or glycogenesis, insulin activates glycogen synthase by dephosphorylation. Glucagon and epinephrine and AMP activate glycogen phosphorylase by phosphorylation, right? So that should make sense to you now. When glycogen is broken down by glucagon, glycogen phosphorylase, what does glycogen phosphorylase do? It takes a phosphate and it cleaves the 1,4 glycosidic bond. So over here, it will use a phosphate and put a phosphate onto this glucose, for example, and break this glucose off. All right. So you're not left with the glucose itself, you're left with glucose 1-phosphate. So glycogen phosphorylase will break the glucose off the glycogen chain by using a phosphate group, and it will attach that phosphate group onto the glucose. So you form glucose 1-phosphate. It can only do this again for alpha 1,4 glycosidic bonds. At branch points, it can't work. So glycogen phosphorylase starts removing the glucose molecules one by one. As soon as it reaches the branch point, it can't break the alpha 1,6 linkage. And so for that, you need debranching enzyme. All right. So synthesis, for glycogen synthesis, remember glycogen synthase and branching enzyme. For glycogen breakdown, remember glycogen phosphorylase and debranching enzyme. Now you've generated glucose 1-phosphate. Glucose 1-phosphate gets converted into glucose 6-phosphate. It's a low yield enzyme. You don't need to know it. Now in the muscle, glucose 6-phosphate will be used for ATP because the muscle is selfish. It requires ATP. In the liver, and in the liver only, glucose 6-phosphate is converted to glucose by an enzyme called glucose 6-phosphatase. This enzyme is only present in the liver, right? This is very, very important to know, because that means that glucose 6-phosphate in the liver and glycogen breakdown in the liver can be used to make glucose, and that glucose can then go out into the bloodstream to maintain blood glucose. So the function of liver glycogen, of glycogen in the liver, is to maintain blood glucose. That's because the liver has an enzyme called glucose 6-phosphatase, which allows it to break down glucose 6-phosphate to glucose. The muscle doesn't have this enzyme. So when the muscle breaks down glycogen, the end product of glycogen breakdown in the muscle is glucose 6-phosphate. This can't go out into the blood to maintain blood glucose, right? So the muscle only has one option, which is to utilize that glucose in ATP production. The liver, on the other hand, has the option of either utilizing it for ATP or using this enzyme to convert it to glucose, send it out into the bloodstreams to maintain blood glucose levels, right? So we discussed this slide that the sites of glycogen storage are the skeletal muscle and liver. Now you know that the uses of glycogen in each is different. So in the glycogen in the skeletal muscle is used for ATP for the skeletal muscle itself. Glycogen in the liver is used to maintain blood glucose. All right, this is important for you to know. So we're done with glycogen synthesis. All right, now there are certain diseases which cause abnormal glycogen breakdown. So glycogen breakdown is ineffective in these diseases. As a consequence, glycogen would accumulate. Glycogen accumulates. That will cause an increase in water coming into the cell because glycogen is osmotic. That causes the cell to swell up. And that's why in these diseases you see liver enlargement, hepatomegaly, you can see enlargement of the heart or cardiomegaly, right? So there are 6 diseases. You all should know this one, right? You all should know this disease here, Pompe disease, which is type 2 glycogen storage disease. So now, let's go back to this slide. All of this stuff is happening in the cytoplasm, right? In Pompe disease, there's an enzyme called alpha-1,4-glucosidase that's found in the lysosomes. No one knows the function of this enzyme. We don't know what it does, how it contributes to glycogen breakdown. But we do know that if this enzyme is defective, it causes Pompe disease. And Pompe disease manifests with enlargement of the heart, cardiomegaly. If you see cardiomegaly on your exam, always choose Pompe disease, okay, for MOL 1. So that's one symptom of Pompe disease. The second one is hypotonia or reduced muscle tone and death usually by 2 years of age. We don't know what this enzyme does, but we know when it's defective, it causes Pompe disease, and that causes cardiomegaly, hypotonia, and death by 2 years of age. Okay? So a summary of this lecture would be that you should remember that glycogen is stored mainly in the liver and the muscle, but its purpose is different in both organs. In the liver, it's used to maintain blood glucose by virtue of an enzyme called glucose 6-phosphatase, which allows glucose 6-phosphate to be converted to glucose, and that glucose can go out into the bloodstream. In the muscle, this enzyme is absent, and so in the muscle, glycogen breakdown is just used for ATP production. Okay, glycogen synthesis or glycogenesis occurs in a well-fed state, activated by insulin, which activates glycogen synthase by dephosphorylation. Branching enzyme is needed for alpha-1,6 linkages. All right, so as we discussed, glycogen is a branch structure. Glycogen synthase, which is activated by insulin by dephosphorylation, causes the formation of alpha-1,4 glycosidic bonds. Branching enzyme forms alpha-1,6 linkages. Glycogen breakdown occurs in fasting conditions or in exercise. The rate limiting step is glycogen phosphorylase, which is activated by glucagon, epinephrine, and AMP. These mediators or these hormones, these basically cause phosphorylation of this enzyme, which activates it, right? For breakdown of branch points or breakdown of alpha-1,6 linkages, you need debranching enzyme. For the one glycogen storage disease, you need to remember is deficiency of lysosomal alpha-1,4-glucosidase. It has an unknown function, but we know that when it's defective, it causes Pompe disease, which manifests clinically as cardiomegaly and death by 2 years of age. Okay, this is a question for you guys. So this was a 52-year-old man undergoes an exercise stress test for 1 week, history of substernal chest pain. So again, when he's exercising, he has pain. During the test, there is a substantial increase in the breakdown of glycogen in his muscle cells. Which of the following changes best explains this intracellular finding? What do you guys think? So glycogen is being broken down. So this question is asking you what enzyme basically mediates glycogen breakdown? Does glucose 6-phosphatase mediate glycogen breakdown? No, this enzyme is present in the liver, which allows it to maintain blood glucose by converting glucose 6-phosphate to glucose. All right. Decreased activation of protein kinase A. That's, I think, a bit above your level. Glucagon activates protein kinase A. And we know that glucagon causes the glycogen breakdown. So if glucagon is active, this enzyme would be increased, not decreased. Activation of glycogen phosphorylase. This causes glycogen breakdown, so this is the correct option. This is the next question. A 7-month-old infant is brought to the physician because of a 2-month history of fatigue and weakness. He has difficulty feeding. Physical examination shows pulmonary edema. All right, this is pulmonary edema, and no hepatomegaly, no liver enlargement, and decreased muscle tone. When they got a chest X-ray, they saw enlargement of the heart or cardiomegaly. What disease is this? This would be Pompe disease caused by a defect in lysosomal alpha-1,4-glucosidase. Another name for the enzyme is acid maltase. All right. So this is just another name for alpha-1,4-glucosidase. Its defect causes Pompe disease, which manifests as cardiomegaly. If you see cardiomegaly on this exam, put Pompe disease or alpha-1,4-glucosidase deficiency. That's it. Thank you very much. Any questions? Okay. Thank you. I think we should